Okay. 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 Hello, my presentation is on sleep deprivation in adolescent mice impairs long-term memory till early adulthood via suppression of hippocampal aerocytes or astrocytes. This was done by Ji Yun Kand, Kin Siak Lee, Jin Hua Wand, and Chiang Guo Sun in 2024. Sleep by Oxford University Press, Sleep Research Society. This was, or the location of this research is Daejeon Hospital of Daejeon University, or Daejeon, South Korea, or in Daejeon, South Korea. My name is Christopher Ranyag. I am in section 01, or section 1 of mammalian physiology. 342. Okay. So this study mainly focused on the physiological system of the hippocampus. This region of the brain is responsible for restoring long-term memories, specifically neurogenesis, which is the creation of new neurons, and astrocytes, which are glial cells that support neurons. They release growth factors and regulate waste. This study primarily focuses on adolescence, which is a crucial period of brain maturity. Modern teenagers commonly get insufficient sleep, which is linked to poor learning, anxiety, and cognitive problems. The point of the study was to provide experimental evidence that sleep loss in adolescence causes lasting memory impairment that continues onto adulthood. This study relates to what we learned in class as memory formation, and the hippocampus is under the nervous system. Yeah, under the nervous system. And neuroplasticity, sleep, glial cells, and stress are all topics that were covered. So, does sleep deprivation during adolescence cause long-term memory impairments that persist into early adulthood? And what are the brain mechanisms that are responsible for these effects? Moreover, they were trying to determine which specific hippocampal pathways cause cognitive deficits, such as hippocampal neurogenesis, suppression of aerocytes, even molecules such as BDNF and AQP4. This question is relevant as adolescents are commonly experiencing chronic sleep deprivation and restriction, but long-term biological consequences are not well understood. With this, we could explain cognitive issues that are linked to poor sleep, and possibly find therapeutic strategies or maybe even emphasize sleep importance. So, mice were the mammal studied in this experiment, and they were studied during adolescence, postnatal day 28, which is equivalent to teenage years in humans. The experimental design was 14 days of sleep deprivation, which is about 20 hours per day, on mice that are 28 years old, or days old. And this was done with a modified multiple platform, which basically just disrupts the REM sleep. 2 points were tested, the postnatal day 42, adolescence, and postnatal day 85, which is early adulthood. And normal mice were used as controls. So the key measurements and techniques that were used was behavioral tests, which test memory and activity. We used a passive avoidance test, which is long-term memory, where mice learned to avoid dark chambers, which gave them a foot shock. Novel object recognition test, which is recognition memory. They just have objects. If they spend more time with it, then it shows that they have more memory or better memory that attributed to that item. And then Y maze, which is short-term spatial memory. It's a maze with three arms, and the mice tended to have a new, or mice who tended to explore new parts of the maze were seem to have better memory than those who just stayed familiar with the familiarity of the ones they've already been to. Open field test, which is activity and anxiety. It measured anxiety behaviors. If they stayed near the walls, they're more anxiety based. If they were more in the middle, it shows that they're more less anxiety. They want to explore more. And more distance means that there was greater activity. More distance covered was greater activity. Biological measurements were also tested. They measured hippocampal neurogenesis, where they used immunofluorescence, such as markers of BRDU and DCX, which are newly formed neurons, and NEUN, which are mature neurons. Western blotting was used for protein measurements, used for aerocytes and neuroproteins, and ELISO was used for measurements of concentration of molecules used for cytokines. The mice were also dissected, and the hippocampal region of the mice's brain was tested for abnormalities. I chose figure two for its importance in behavioral changes in sleep deprivation and directly showing the main result of the paper, which is that sleep deprivation impairs long-term memory, but not short-term memory. The figure demonstrates that sleep deprivation or sleep deprived mice had worse long-term memory, both in PND42 and PND85 mice. The figure shows that there was lower passive avoidance or for that test, there was sleep deprived mice had a much shorter latency time, indicating worse long-term memory. The SDA mice in novel object test had a much lower discrimination value, indicating they explored the new objects less, and lower ratio, indicating worse recognition memory. The Y test indicated no significant difference between groups at either age, indicating short-term memory remained normal. An open field test showed that there was no significance um, in the difference of time spent in the center zone, showing similar anxiety levels between the groups. The mice traveled significantly more distance in PND42 in the open field test, showing mild hyperactivity after sleep loss, but PND85 activity levels normalized. Sleep deprivation significantly impaired long-term memory, but not short-term memory, as seen in the Y maze test. And long-term memory impairments lasted well into adulthood, even after sleep recovery in PND42 and PND85. SDA mice temporarily are more active in PND42, but activity or anxiety does not change. This all points towards to how adolescent sleep deprivation damages the hippocampus, which is responsible for long-term memory. Okay. So for figure three was chosen as it shows the mechanistic results of the experiment through Western blot and immunofluorescence. It shows how sleep deprivation affected neurogenesis in the hippocampus, and the Western blot of immature developing neurons, DCX, and mature neurons, NEUN, both showed lower bands in sleep-deprived mice. Sleep deprivation reduced new and mature neurons in the hippocampus. DCX and NEUN are relative to alpha turbulin. Both are significantly lower in SDA mice with a p-value less than zero point 0.1, and in both PND42 and 85. It's like that. Immunofluorescence showed that SDA mice had a GCX green yeah. So DCX, which is green, had fewer immature neurons. BRDU, which is blue, show that which is also an immature neuron, had fewer newly dividing cells, and NEUN, which is red, had fewer mature neurons. This was taken from the dentate gyrus, which is the main side of the hippocampal neurogenesis, and the CA1 region of the hippocampus. Finally, panel D shows quantification of immunofluorescence, confirming significant decrease in DCX, BRDU, and NEUN positive cells. It is very strong significance with a p-value of less than 0.01. Neurogenesis is reduced both immediately after sleep deprivation and long after recovery, showing the effects do not reverse on their own, and sleep deprivation during adolescence significantly reduces hippocampal neurogenesis, which these deficits persist into early adulthood. Okay, so the key findings was that adolescent sleep deprivation caused significant long-term memory impairments, which is shown through passive adolescence and novel object tests, while short-term memory remained unaffected. This indicates vulnerability of long-term memory systems. Hippocampal neurogenesis was reduced, shown by the lower DCX, BRDU, and NEUN neuron levels, and these structural and molecular deficits due to SDA persisted into early adulthood, even after 6 weeks of normal sleep to recover. This tells us that the hippocampus is highly sensitive to sleep deprivation during development, and a reduced neurogenesis means that there are fewer new neurons to support memory function. Sleep during adolescence is critical for healthy brain development, as young brains are impressionable, and sleep deprivation causes long-lasting functional deficits. Connections to this class is that it connects to what we learned about the nervous system and how it functions. Hippocampus is essential for memory consolidation, and neurogenesis and neuroplasticity depend on a proper healthy brain. Sleep promotes synaptic strengthening and circuit formation, and REM sleep is important for memory and repair, and we should aim to get lots of deep REM sleep. Critical analysis and future directions is that the study limitations were that there was no comparison for adult sleep deprivation. The study only tested sleep deprivation in younger mice, and it's unclear for adults if they experience the same. Limited sample size, as the results were based on pooled samples of small n values, and that could just reduce precision. This article only explored up until early adulthood, and deficits like continuing to mid and late adulthood is just unknown. What could be improved? More groups could be added, adult sleep deprivation could be tested, and different durations or intensities of the type of sleep loss. We could use alternative sleep deprivation methods to minimize stress, not just REM sleep. And we could go into more precise subtypes of protein formation or genetic tools to identify what specific functions of protein synthesis were disrupted, and collected more long-term follow-up data, or we could also collect more long-term follow-up data on the mice to see if the brain ever fully recovers. So for future research directions, exploring restorative conditions such as boosting AQP4 or BDNF astrocytes, and if it's possible to reverse memory deficits. Any tests to possibly reverse neurogenesis for memory recovery, testing to see if adolescent sleep loss affect the brain later in adulthood, not just early, and lastly, to find if adolescents are uniquely vulnerable, testing adults versus early childhood mice. And these are my references. Thank you. Have a great day.